Everyone. So I'm the last hurdle before the break and coffee and refreshments. So thank you for attending. Excellos 360 describes a platform we've developed to try and improve the quality of the cell therapies that are being used in the clinic, both CAR T, TIL-based, as well as other adaptive immune systems. Who are we? We're a CDMO serving industry needs with customized development and manufacturing capabilities. We're very nimble. We realize that right now the cell therapy field, in particular the manufacturing process development part, is still fairly plastic and dynamic, and so we afford that type of flexibility, service, and capability to the clients that come to us with their specific needs for their cell therapeutics, including starting material, and I'll focus a bit on that today. Manufacturing capabilities, we have CGMP compliant clean room spaces for manufacturing under regulatory auspices, and extensive capabilities for characterization and manipulation of the cells. In the clinic, the issue is variability. And by that I mean you can take a given therapy, whether it be a molecular-based, cell-based, or whatever. You try to stratify patients to get a relatively homogeneous intent to treat population, but still you see a variety of responses, from no response whatsoever, progressive disease, all the way to complete responses. And there are three main factors that drive that: the innate biology of the tumor and the host themselves, the patient's status. That's, if you will, force majeure factors that we can't really control. You get what you get when the patient presents with their disease. There's also inherent tumor-resistant mechanisms. We've all heard about PD-1, CTLA-4, and other intratumoral microenvironmental suppressors of the immune response to the cancer. But a third one that we think can be addressed even better than we currently are doing so is product variability. And our approach is to carry out deeper cell characterization and implement that aspect in our quality by design assessments to decrease variability and improve final product quality in support of better clinical outcomes. The more consistent the product, the higher the cellular API content of the product, intuitively, the less variability you have in the dose that you're giving and inherent, and that would be more consistent clinical results, with the caveat of the other two factors that also cause variability. And here's what we're talking about. All of our dream here is to cure cancer. And I've been in the business for over 30 years, started at MD Anderson. And we made incremental steps forward. People live longer, they have better quality of life, less problems with the disease, but ultimately this has come to it. The cell therapy field has the audacious aspect of it of possibly being able to cure cancer if we do it well. We've already seen some isolated instances of that with some of the CAR T therapies. One study on the left side panel is the diffuse large cell B-cell lymphoma patients that had a CAR T therapy showed that if they had complete responses, and they had an 80 to 90% probability of surviving beyond 12 months, but if they had partial responses, their life expectancy was less than a year, less than 12 months. On the other panel, this is a TIL company's data set. We actually participated in this, supplying them with the feeder cells to generate the TILs. And what they showed here was that if you had a CR, survival is beyond 10 years right now. If you had a PR, partial response, which means 50% or at least 50% reduction in tumor burden, you had about a 22 to 24 month survival probability. So obviously, we need to flip that ratio of PR to CR to a higher percentage of complete responses with cell therapies to do a better job in the field. Our approach that makes us a little bit different from other CDMOs is that we have focused for the last couple of decades now on the CAR constructs themselves, and we're now up to generation 7 for a CAR construct for CAR T cells. And every one of them has made incremental improvements in the efficacy and the functionality of the cells. But especially now that we're moving into the allogeneic space, the starting material plays a role as well. If your cells are not high performing in terms of the other downstream pathways downstream of the CAR construct signaling system, you may have issues with the potency of the product. And we focus on characterizing the starting material because it's our belief that garbage in, garbage out plays a role in product variability and lack of more robust responses. The FDA actually bemoaned that. They said the nature of the cells used for as starting material may be critical for CAR T cell therapy quality and function. Due to patient donor variability, cellular starting material can represent a major source of lot-to-lot variability. That was a white paper they had out, basically asking us all to do a better job of characterizing the material. And we're not just talking about yield, viability, and the right cell type. We're talking about what can the cells themselves do. What innate potential do they already have? And our approach is to identify superior starting materials to assure better potency. Inherent in that would be the reduction in product variability and the improvement in quality of the final product, which hopefully will enable more consistent clinical results and more robust clinical results. And our methods are as follows. We do immune cell profile screening of the donors for allogeneics, healthy donors, just to see what the repertoire of cells looks like. There's no one donor fits all needs for the adaptive immunotherapy space. We also look at the metabolic and effector potential of the cells, of the starting material, to assure that the cells have the right weaponization potential and also durability in terms of metabolic fitness. And then we'll use those types of assessments to look at manufacturing intermediates. After each major manipulation of a cell, are you affecting the potential of the cells, the potency of the cells, and the properties of the cells that you need for an optimal therapy? We are also looking at, by quality, using in our QBD assessments of platforms, do different platforms impact these properties? Because some of these systems that we use to enrich and isolate the cells are pretty rough on the cells themselves. And this is just a snapshot of some data showing that when we look at immune phenotyping by FACS analysis, that you see quite a bit of variability in normal healthy donors in terms of their immune cell repertoire, whether it be CD3, 4, 8, 56, and we've done it for other subsets as well. There are some trends for donor age to correlate with decreases or increases in certain subsets. And one could argue, well, let's just take young donors and avoid the variability. But as you can see, even within young donor cohorts, there's quite a bit of variability there in terms of the repertoire. Effector function. Do the cells have the weaponization needed to kill the cancer? There's a proteomics-based platform developed by Isoplexis called the Isolite system that uses a 32 proteomic signature to develop a scoring system called polyfunctional strength index. That PSI index now has been shown in multiple clinical studies to correlate with response or lack of response. If you have a high PSI, as is shown on the right slide in a paper that was published in Blood in a heme malignancy, you see responses. If you are a non-responder, you have a low PSI. The nightmare is that when you take a series of just normal donors and look at their PSI index, it's quite variable, and that's depicted on the left panel side for 7 different donors. So what we've done is we've built in an assessment of the PSI potential of the starting material as a way to reduce the risk of picking low-performing cells that you're going to use as a CAR T, TCR, or other type of adaptive immunotherapeutic. We've done now up to 50 donors looking at the PSI potential and effector potential of the cells. And you can see with age, there seems to be a trend for a decrease in potency. No surprise there. But even within certain data strata, there are age strata, there are still variability even in young donors. So you want to try and pick donors that are not low PSI functionalities, what we believe. And the 2 blue dots in the leftmost panel are just 2 donors we picked with relatively low and relatively high PSIs, and I'll tell you a bit more about them later. And that's the pan CD3s, all the total T cells that we chose. Metabolic fitness. Janet Price at WashU did some beautiful work along with a lot of other people showing that in order for T cells to go into effector function and to kill, they must go into glycolysis. 1, they need the extra energy. But 2, GAP dehydrogenase, which is mobilized during the glycolytic cycle, serves as a signaling system that helps drive the bioactive cytokines necessary for cell killing to be produced and released into the extracellular environment. So it's not just an energy source, it's a protein modulation system when glycolysis occurs. So if you don't go rapidly into a glycolytic state, your effector function is going to be compromised. Also, the durability of the cells in terms of hanging around and having enough stamina, if you will, biological stamina, to repeatedly kill is going to be compromised if they can't go into glycolysis effectively. We looked at the glycolytic status of normal healthy donors, and the variability is startling. And this is just an exemplary data showing that if you want to pick donors, you would try to pick donors with a high glycolytic response index, because that drives better effector function versus donors that are low. And once again, getting old is not a good thing. Not too surprising, you know, as old as I am, I can't run as fast as I could when I was 20 years old. Okay, so we've developed a scoring algorithm using, we're making it numerical now, but right now, just for this talk, we just score donors as low and high. And you'll see two blue dots again. Those are the same donors on the other slide that had low and high PSI values, and these had low and high glycolytic potential values. We took those donors, and here they are. There's their age and their AUC and their PSI, and we ran them in two different assays in a blinded fashion. We sent them to a third party that's agnostic to us and doesn't care about us one way or the other. Said, take this vial of cell, these two different vials of cells, and run them through canonical cell killing and cytokine release assays. And what we showed when we looked at both PBMCs and pan T cells is that the high-performing donor had much more killing efficacy than the low-performing donor, that's shown here. And also, when you look at pan T cells themselves, you just enriched for CD3s, once again, the high-performing donor had much better killing activity than the low-performing donor. So, you're a patient that's going to receive an allogeneic CAR T therapy. The company is going to pick an allogeneic donor to make your CAR T cells. Which donor, based on this data, would you think might be the better one to start with as starting material? Okay? That's the context here. And this is the cytokine release study. See if you can guess which donor was a high-performing donor. This is gamma interferon release. What we're showing is donor 1, the high-performing donor, once again, had anywhere from 200 to 400% higher gamma interferon release as well. These two assays are oftentimes required as potency release assays by the FDA for final product for you to go to the clinic. So we wanted to not just say the phenomenon of what we see in our Excellos 360 platform matters, but that it's linked causality-wise to actual potency assays used for product release. And that also these assays, the PSI index assay, and now more recently, the metabolic assessment, have been shown to correlate with clinical response or lack thereof based on the degree or lack of degree of responsiveness and scoring in those assays. We're probably one of the first CDMOs, if not the only one, that's doing this as an inherent part of donor characterization. The donors that we identify are repeat donors, that we call them high compliance donors. We score them for their willingness to come back and be repeat donors for product. And we have developed a good cadre of these folks. We've had them around for 8 years. The company spun out of San Diego Blood Bank, and we use some of these donors now for 8 years. And we monitor them every time they come in years later to make sure as they get older, things aren't changing. We also looked at those same donors in terms of proliferative outgrowth of a rare subset of cells, gamma delta T cells. We did this for a client we were working with at the time. And what we showed is that, once again, the high-performing donor had much greater fold expansion of a gamma delta T cell subset than the low-performing donor. So it's not just effector function, it's not just metabolic fitness in terms of durability of effect from a therapeutic standpoint, but it also may play a role in the expansion protocols you have to develop to generate enough of the product to engineer to use in the clinic. We looked at different platforms, QBD. We did quality design assessment of some of our platforms. We use a standard manual isolation versus a process that uses differential centrifugation in one case, and a process that uses microfluidics-based enrichment of PBMCs in the second case. And we looked at yield and metabolic potential. What we showed was that, and one of these processes is now standard used in the industry for initial enrichment, okay? And what we showed was that the process 1 had a significant attenuation on metabolic potential. We know why. We figured out why in collaboration with another group. Whereas the process 2, which lacks one of the steps that process 1 does to compromise mitochondrial function, doesn't do that and preserves metabolic function much better. And that's kind of worrisome when you think, well, let me just grow the cells, let me just isolate them this way or that way, and they'll be fine. No, they are not. Not always. So when you do QBD, don't just look at yield and viability. You might want to look at functionality. And that's what we offer as a CDMO to our clients to make sure that if, and part of process development and what platform options you have, which ones might better serve the needs that you have and ultimately the needs of the patient, which are the number one priority here. We also looked at PBMC and PAN-T yield. PBMC yield was slightly better with the microfluidic-based system as well for total peripheral blood mononuclear cells, but what was really interesting was that the T-cell yield was substantially higher. And this is 24 and 48 hours post-thaw after cryoprocessing. So we don't just take the cells and immediately look at the cell count. We cryoprocess them, then we thaw them out, put them into media at 24 and 48 hours and assess what's left to see what grew out well or started to grow out well.
Thank you. We are at time now. Apologies.
Okay, in closing, we have developed a preferred donor identification strategy. In-process monitoring is enabled by this to ensure continuity of desired characteristics. And you can build these assessments into your QBD assessments of various isolation and production platforms to increase the probability of clinical trial success, to improve the clinical outcomes, and give you more consistent product infusion to the patient to get rid of that variability. Thank you.